The figure here is a concept map for blood transfusion. Group O can donate blood to itself and to all the other groups, Group A, B, and AB. Therefore, it is referred to as a universal donor. Group A can donate blood to A and to AB. Group B can donate blood to B and to AB. AB can donate blood only to itself, but it can receive blood from all the other groups. Therefore, this group AB is referred to as a universal receiver. How can we determine the possible group of an individual? As you know, before any blood transfusion, it is very important to determine the blood group of the receiver and that of the donor. This is vital to prevent any complication in the blood transfusion. This technique in determining the possible blood group is called blood grouping. Two different techniques can be used. Let us have a look at the first technique. Here on a slide, the blood of the individual we're going to test is mixed with different test sera containing anti-A antibody, anti-B antibody, anti-A and anti-B antibodies. This you can observe and document in your book on page 117. The blood of the tested individual is going to be mixed with each theorem, and the result is going to be observed agglutination or no agglutination. If agglutination happens with anti-A and anti-B, this is going to signify that the red blood cells of the individual has antigen A. Therefore, the blood group of that person is going to be Group A. If agglutination happens with anti-B and anti-A and anti-B, this is going to signify that the individual has antigen B, which has bound with the anti-B in the theorem. Therefore, that person is going to be of Group B. However, if agglutination happens with all the theorem as you would observe here, this would mean that the blood group of the individual is going to be AB, since that person has both antigen A and antigen B that has bound with the anti-A and anti-B, causing agglutination. Finally, if the blood cells of the individual does not show any agglutination with none of the theorem, this is going to imply that the red blood cells of that individual has no antigen, and that would make that person of Group O. Therefore, as you have observed, the red blood cell agglutinogens, that is the antigens, can be identified by this way, and the blood group can be therefore determined. Another way used to know the blood group is to look for the theorem agglutinin, that is, to look for the antibody in the blood. Here, the test theorem is going to be reacted with control red blood cell of known blood group, that is AB, AB, or O. This is the answer to probing the activity, by the way, number 5 in your book. In the first way that we had previously talked about, we were trying to identify the antigen in order for us to know the blood group. Here we are going to look for the antibody to know the blood group. We're going to take the theorem of the individual, and it is going to be mixed with red blood cells that are standard, that is, that are known. The red blood cell with antigen A, red blood cell with antigen B, red blood cells that have both antigen, and red blood cells that have no antigen. If the theorem shows no agglutination at all, here, it means that the theorem has no antibody, has neither anti-A nor anti-B. Therefore, which theorem has no antibody? It is the theorem belonging to group AB, as you would observe here. The next one, when agglutination happens in with red blood cell having B antigen and red blood cells having AB, it shows that the theorem should have anti-B, but it has no anti-A since it didn't show any agglutination here with the antigen A. So this individual is definitely going to be of group A. In the third case, agglutination happened with A red blood cell having antigen A and red blood cell having antigen AB. The presence of agglutination here is going to indicate that the theorem has anti-A. But the absence of agglutination with antigen B would reveal that the theorem has no anti-B. So what theorem has only anti-A? It is the theorem belonging to group B. In the last case, agglutination is observed with A, B, and AB. So this is going to imply that the theorem here has anti-A, which has caused agglutination, the plus here by the way, with A antigen, that is antigen A, and it has anti-B antibody that had caused an agglutination reaction with the antigen B. Of course, agglutination reaction happening with the red blood cell having A and B, but no agglutination happening here, of course, since O has no antigen. Therefore, the blood group that has both anti-A and anti-B is going to be that of group O. Another self-marker found on the red blood cell is the rhesus factor. Apart from the ABO system, the rhesus system is the second in importance in the immune responses. Know that there are at least 30 commonly occurring antigens have been found in the human red blood cell, but most of them are weak and therefore are of no importance in immune reactions. An individual is said to be rhesus positive or rhesus negative. On what basis is an individual classified as Rh positive or Rh negative? Let us have a look at the adjacent document, which clarifies the notion. This is the red blood cell, and on the red blood cell we have a certain marker, which is the Rh factor. The presence of the Rh factor is going to cause the individual to be Rh positive. Obviously, The absence of the Rh factor on the membrane of the red blood cell is going to render the person Rh negative. By the way, the Rh is so called because it was first found in the blood of rhesus monkeys. The second figure clarifies the notion concerning the ABO system and the rhesus system. As you would observe here, this is the red blood cell of an A positive individual. The A positive individual is going to show on the membrane of the red blood cell the antigen A of the ABO system and the rhesus factor. The antigen for the rhesus factor is here. So this is why this person is referred to as an A positive individual. On the other hand, an A negative individual is going to have on the membrane of the red blood cell the presence of the antigen A only. This individual shows no rhesus antigen, that is, no rhesus factor. This is why he is called an A negative individual. The allele Rh positive is dominant with respect to the allele Rh negative. So, for a person to be a rhesus positive individual, he's going to have the two genotypes shown here. He's going to be homozygous Rh positive or in a heterozygous form Rh positive, Rh negative. And for a person to be rhesus negative, he's going to have this genotype, that is, in a homozygous form, Rh negative, because, as you know, a recessive trait will only appear in homozygous form.